Okay, well, maybe we'll go ahead and start. Um, so please, if you haven't already signed in, please sign in, and I will try to remember to remind you again. And we are going to be doing some hands-on work. I know that some of you I've seen before, so probably most of you shouldn't have any problem getting into the DCC on-demand system, but if you have not, please follow the instructions in this link and that Richard put in the chat to be sure you can get in. And if you have a problem, we can try to fix it between now. I'm going to be giving a lecture for probably the first for 45 minutes or an hour. So if you have a problem, we can try and fix it between now and then. And otherwise, all the material will be available offline. And actually, speaking of that, so just in case you are haven't been here before, all of the course material is all the workshop material is available at the link that I just put in from GitLab. Okay, so I am here to talk about bulk RNA-seq. Just going to set myself a timer so that I remember to take a break. I'm going to aim to take a break around 10 o'clock and around 11 o'clock. Just short breaks, but so that you can have a minute away from Zoom. Hopefully refresh mentally. Okay, so I'm here to talk about bulk RNA-seq. And I'm specifically going to be talking about the bioinformatic aspects of this. Next week, I'm going to be talking about the theory and some bioinformatics for single-cell RNA-seq. And then in 2 weeks, Dr. Shi is going to be talking about the statistical aspects of analysis for bulk RNA-seq. And then in the 2 weeks after that, Tyler Shapey is going to be talking about more of the data visualization statistics for single-cell RNA-seq analysis. So this is like kind of an intro to some extent for a lot of material, really what we're going to be covering for the rest of the semester. And so a lot of the stuff that we talk about today is actually, relevant to single-cell RNA-seq. Single-cell RNA-seq adds on a sort of level of complication. So I'm guessing everyone here is somewhat familiar with RNA-seq. But just as a sort of quick motivation, I just want to talk briefly about like a few different ways that it can be used. The most common way for RNA-seq to be used is to do transcriptome analysis. So looking at all the protein coding genes that are being expressed in a sample. I guess really it could be all the genes that are being expressed in a sample. So basically all the RNA that's being made in a given sample. And then the most common application for that sort of data is looking at differential expression. So asking, okay, I have sample 1, which is my control, and sample 2, which was received some treatment, maybe with a drug. And I want to know what genes are being expressed at a higher level in my treatment sample than in my control sample, and what genes are being expressed at a lower level in my treatment sample than my control sample. And, I can get more detailed. I can say, like, okay, what's the relative change in expression level, things like that. Also, transcriptome data can be used to annotate genomes. So if you're working in an organism where you've sequenced the genome or not, and you want to know just like where the genes are, there are computational ways to do that if you have a genome sequence, but those are error prone, and transcriptome data does a much better job of helping you annotate genomes, with finding where, really in combination with computational methods, finding like where there are genes in the genome. You can also use RNA-seq data to identify sequence variants. That is also something that can be done with DNA data. And also to detect gene fusion. So this is where you have a structural rearrangement in the genome so that you get genes put together that aren't normally together. And these are actually pretty important in a tumor, both in researching cancers, but also they're used to some extent clinically in identifying treatments for different cancers. And so what we're going to be talking about today is going to be almost entirely focused on differential gene expression. And just a little bit, I already touched on this before, but just you can divide RNA-seq. There are different ways that you can divide RNA-seq data, but one way to divide it is between bulk RNA-seq and single-cell RNA-seq. And I already told you I'm going to be talking more about single-cell RNA-seq next week, but just to be clear, bulk RNA-seq is when you're taking a sample. Usually you're talking about on the order of at least 1 million cells. It could be more, it could be less, but you're taking a collection of cells in a sample, and you're grinding up all those cells, and you're extracting the RNA from them, and then you're sequencing. So what you're getting is an ensemble of the RNA from all the cells present. And depending on how you prepare that sample, that could be a mixture of cell types. Like, for example, going back to what I mentioned before, the tumor. If you grind up a tumor, tumors are heterogeneous tissues. Most tissues in an animal are heterogeneous, but tumors are a particularly good example because you have some tumor cells, you have some normal cells, you have some immune cells in there, and when you grind up the tumor, you're putting everything in a mix together, and so you can no longer directly identify all the different cells in that mix. You're going to get an average information that's an average of all the cells in that population. Single cell RNA-seq, the goal is to actually be able to look at individual cells in the sample and say, okay, this cell is, I have one cell here that has this RNA in it. I have another cell that has this RNA in it. I have another cell that has this RNA in it. And so you can actually, you're not treating the sample as this, you're treating the sample as individual cells. And there's some big benefits of doing that, but as I'll talk about next week, there are also some negatives of doing that. So there are places for both bulk RNA-seq and single cell RNA-seq. And again, this week I'm going to be talking exclusively about bulk RNA-seq, but like I said, understanding bulk RNA-seq, I think it's really I'd say essential for understanding single-cell RNA-seq, because single-cell RNA-seq is just kind of, you're adding a little bit of a layer of complication, but a lot of what we're going to be talking about this week, we're going to build on next week when we talk about single-cell RNA-seq. And I should say, actually backing up, nope. Backing up, please feel free to interrupt me if you have questions. I will try to keep an eye on the chat, but sometimes it's hard to see what's going on. And yeah, and if I missed you, you're also welcome to just unmute and ask questions if you want. Okay. So in a nutshell, this is bulk RNA-seq in a nutshell in terms of the analysis. I'm going to talk about, oh no, actually I talked last week. I talked a little bit last week about how we actually prepare the samples. So what I'm going to be talking about this week is almost entirely about how we analyze the data. And so this is, in a nutshell, what the data analysis looks like. And again, I'm talking about the bioinformatic aspects of this. Dr. Shi will be talking about the what you do after you've done this bioinformatic analysis. And so the input to the analysis that we're doing today is sequences of RNA fragments. And then the output from what we're doing today is counts of gene fragments. So what that looks like sort of a little bit more practically is, here's a little of a snapshot of sequence file. So this is in a FASTQ format. We'll talk a little bit about what that means soon. But you can see here, if you look closely here, there's A, T, C's, and G's. So that's actually sequence from RNA. And so the data sets that we're going to be working with, we're going to have, on the order of millions to billions of these individual sequences. And our goal for today is to take that data and essentially summarize it into a table that for each sample, we're going to want to ask for each gene in the genome, how many times we saw a sequence, an RNA sequence fragment from that gene, right? So here, this is sort of just a toy table. For gene A, we saw it 5 times in the sample. For gene B, we saw RNA 11 RNA fragments, 11 RNA fragments in the sample. For gene C, we didn't see any, and for gene D, we saw RNA fragments, 2 RNA fragments in the, right? And so basically, if you fall asleep for the next 2 hours and 40 minutes, this is what we're doing today, is going from here to here. And so I'm going to talk about kind of why and how to think about this, and we're going to do this hands-on. But this is basically our goal for today. Okay, so I like to think about what's happening with RNA-seq data with this analogy. And I use this analogy of I call it a biblioarchaeologic analogy. And so you can imagine that there's someone, 20,000 years in the future who is doing archaeology in this region of North Carolina. And they know that 20,000 years ago, there were these, there are these myths of this great rivalry between 2 major cities, the city of Durham and the city of Chapel Hill. And they want to know, okay, what were people in these cities reading? Maybe that'll tell us about the culture in those cities. And so they've identified the locations of the libraries in these 2 cities, the Durham Public Library and the Chapel Hill Public Library. And they've excavated at those sites, And what they've come up with is fragments of text from books, right? So, here's an example of something they might have come up with. And you can see, you have these like chunks of fragments that, here is a fragment that's not really attached, but you can kind of puzzle piece it together. There's some other big chunks. So just to sort of simplify things, and so again, the goal is to try and understand the cultures of these two cities using these fragments that have been excavated from the library. So just to simplify things, we're going to assume that the fragments are continuous lines because it's a lot harder to work with these like sort of irregularly shaped fragments. So imagine that the fragments, we got lucky and the books just happen to fragment as like whole lines. And also to simplify things, we're assuming that the fragments are, so each of these lines here is a separate fragment. And they all just happen to be, lucky for us, they all happen to be 100 characters long. And when I say 100 characters, I'm counting, spaces and punctuation as characters. And so our goal is going to be to take these fragments and we want to, again, try and understand what people were thinking, what were the cultures of the people living in Durham and Chapel Hill in the year, let's say 2025, based on what books were present in the library. And so we have in addition to this data, we have another resource, which is a database of all the books that were in publication around that time. And not just the names of the books, but also the full text of the books. And so our goal really is to, what we're going to try and do is to try and identify for each of these fragments what book it came from. And then, based on that, try and see if okay, are there differences in books that were being read in these two cities? Everyone, any questions about that so far? Okay. So the first thing that we're going to do is we're going to take each of these fragments and we're going to search it in our database of the texts of the books that were in publication at that time. And then, that'll help us to identify, what the differences in sort of book distributions are between these two libraries. And so you can imagine, if you want to think about like the algorithm for doing that, you can think of a really simple algorithm where you would take, you have this, again, this whole database of all the books that are in publication. And one way to search through this database, we're not going to get like too deep into algorithms, but I just want to give you a little intuition for thinking about how you could do this. So one way you can think about doing this is you would take this N here, that's the first character of the first fragment, and you would search it against the database. So basically you look through each letter of the database, and if the first letter of the database is an N, then you would then go to the next letter of the fragment, which is a G, and say, okay, well, is the next letter in the database a G? And if it is, you keep going. You say like, okay, well, next there's a space in the fragment, so is the next letter in the database a space? And let's say it's not. So then you say, okay, well, it's not matching the first 3 letters of the database. So let's start again at the second character of the database. Is it an N? If not, go to the third character of the database. Is it an N? If you keep on going until you find a character in the database that's an N, and then you ask, is the next letter a G? Right? And we can keep doing this. This is going to be slow and tedious, but if we have a computer, that's going to speed things up for us. And you can imagine you just keep on doing that until you match the whole this whole fragment, had a perfect match to the database. And then you say, okay, well, it looks like this fragment is actually coming from this book that's, that we're searching through right now. Does that make sense? Any questions about that? And so I'll just tell you that's like kind of like a simple algorithm. I think that like anyone who knows a little bit of programming could probably write that algorithm. It's not particularly efficient in terms of speed or other resources, but it would work. If you want to do it faster, they're actually, computer scientists have spent decades thinking up better algorithms to search for given texts in a database of a larger database of text. And so there are faster ways to do that. They're more complicated. I'm not going to go into exactly how you would do that, but you can think that you don't need to worry about the like sort of more complicated ways to do that because other people have figured that out. If you care about that, I can point you to resources for faster ways to do the search. But just imagine, so we're going to take each of these fragments, search the database, and we're going to then label the fragments with the book that the fragment came from. And so that's going to get us to this. And actually, I didn't mention this before, but just for just because we can, here's the book, the title of the book that the fragment came from, and then I'm also indicating the character number that the first character here matches, right? So this N here, if you look in the text of Huckleberry Finn, book by Mark Twain, this character here is character 471,971 in Huckleberry Finn. So, we've done that for all of these fragments. Remember, each of these lines here is a separate fragment. And so we've been able to match all of them. Well, so actually not quite all of them. I'll tell you that when I generated this example, I kind of made a little bit of a mistake, which I think is actually a really useful mistake because we get the same thing with real data. If you look carefully, there are these two fragments here that I marked with question marks. And if you read them, they kind of sound like a little bit more legal text than maybe a novel. And that's because the database that I use to generate this example is from an organization called Project Gutenberg that has like thousands of texts of books that are no longer copywritten. So you can freely download books from Project Gutenberg. It's a really nice resource. And at the beginning of each file, they have a sort of legal statement about the copyright status of the book. And so when I generated this example, I accidentally pulled out some of the two fragments with the copyright text. In the sense of thinking about this as these are good examples of kind of the sort of information you get from real RNA-seq in two ways. One is that every book in the Project Gutenberg database has this text, so we can't actually assign it uniquely to any book because it's gonna, if our database is from Project Gutenberg, then it's going to match every text. So we get this with real RNA-seq data, and there are different ways of handling this, but one is just to say like, okay, we're just going to ignore this because we can't get any useful information out of it. The other thing that this is a good example of is that you always get, when you generate RNA-seq data, you get sequences that are essentially garbage from the perspective of it's not biological sequence, and there are all sorts of or it's not sequence that matches your organism at all, whether it's biological or not depends. And so again, what you tend to do is you just throw that out. Now sometimes that that data is useful in diagnosing problems. And in this case, these examples are useful in diagnosing, when I ran through developing this toy example, I was like, oh, where's this text from? And then I figured out, I know where it's from. So even though we're going to throw these out for the next steps, it is useful to look at them and say, here's something that I didn't expect. Why is it here? And how much attention do I need to pay to it? So I know where these came from. Again, we're going to throw them out for the next steps, but I know why this sort of unexpected result came about. Questions about any of this before I keep going? Okay. So we have, again, like for each of our fragments, we have the book it came from and the position in the book that it came from. And so again, the next, really what we're aiming for is a table like I showed you before, where we, for the library, we figure out how many times we've observed each of the books in the library. So I've thrown out those weird legal texts. And so the next thing to do is we're just going to group these fragments by the books that they came from. So that's pretty easy. We just kind of like sort them by the title of the book. And so you can see, you can eyeball this and just see, okay, well, I have three fragments from Ulysses. I have 3 from Alice's Adventures in Wonderland. I've got 5 from Huckleberry Finn. I've got, I don't know, what is that, 8 or 9 from Tale of Two Cities. But really, like, we do want exact numbers, right? So we can either, I can count these by hand, or I can let the computer do that. And I prefer to let the computer do that. So the computer is going to give me these numbers. And so this is what I want. I want a table of the books and the number of fragments from that book. So this is, pretty much we're done, except I told you that we want to be able to compare between libraries, right? And so really what I want is that table from the Durham Public Library and the Chapel Hill Public Library. And so if I generate that table, I can see, there's some differences here. Now, one thing that I want to point out is that I can look at this and I can compare, and I can say, okay, well, I am seeing 3 times as many fragments of Alice's Adventures in Wonderland in the Durham Public Library as the Chapel Hill Library. Now, again, you're going to be talking about statistical analysis in a couple of weeks, but I just want to mention here with this example that, my sort of naive assumption might be, okay, well, there might be 3 times as many copies of Alice's Adventures in Wonderland in Durham as in Chapel Hill. But I don't know if I have enough data here to like make that, if that's like a real statistically valid assumption. So like maybe, looking at Tale of Two Cities, we've got more data. So like maybe I feel a little bit more comfortable saying that there are twice as many copies of A Tale of Two Cities in Chapel Hill as in Durham. Maybe, that's something that we actually need to do an analysis to figure out whether that's a valid conclusion. The other thing that I want to point out is that if I want to look within a library and say, okay, well, I see that in Durham, I have 3 times as many fragments from Anna Karenina as I do from Alice's Adventures in Wonderland. So maybe I have 3 times as many copies of Anna Karenina in Durham. There's a major flaw with that conclusion. Anyone know what that is? What's the problem with looking at these numbers and saying, okay, well, within Durham, I have 3 times as many copies of Anna Karenina as Alice's Adventures in Wonderland. Someone's guessing in the chat. Yeah, maybe the total number of sentences in these books are different. Yeah, so if maybe Anna Karenina is actually 10 times as long as Alice's Adventures in Wonderland. And so so I'd expect if if there was say 5 copies of each book in the library, I'd actually expect to have if I have 3 fragments from Alice's Adventures in Wonderland and everything else is equal, I'd expect to have 30 fragments from Anna Karenina if it's 10 times as long, right? So I can't maybe make that comparison. Another possibility is that actually the copies of Anna Karenina, maybe Anna Karenina was hardback books and Alice's Adventures in Wonderland was paperback. And so the hardback books, the hardcover on the hardback books actually protected those books from breaking down. And so the books of the Anna Karenina has just lasted longer, or maybe it was printed on higher quality paper or things like, so there are all sorts of things that can contribute to making mistakes in comparing book to book. And so it can be, that is not something that is often done. Well, of course, not necessarily in books, but also with RNA-seq, because you're not necessarily comparing apples to apples if you're comparing different books within a sample. If you're comparing Alice's Adventures in Wonderland in Durham to Chapel Hill, now you don't know, it could be that Chapel Hill and Durham have one of them has hardback books and one of them has paperback books. Let's ignore that for now. It is more likely, we know that the number of sentences is going to be the same between 2 different, copies of the book. And so at least we can make that comparison sort of safely. And we don't have the situation where Anna Karenina might be 10 times as long as Alice's Adventures in Wonderland. If we're comparing Alice's Adventures in Wonderland to Alice's Adventures in Wonderland, we at least have that sort of consistency. Okay, any other questions? Any question about that sort of analogy before we actually talk about real RNA-seq data? Okay, so let's talk about real RNA-seq data. So this is an outline of the steps that we're going to go through today for processing the raw data into that table of counts. And so here's the step. Here's a sort of summary of the input and output. And then each of these steps actually has its own file format, and we're going to be looking at those. So we kind of just went through, we went through some of these steps, right? With the library example, we went through the mapping step and the counting step, and those are going to be pretty similar. We'll talk in a little bit about the sort of pre-processing and quality control. So let's start really quick looking at the FASTQ file format. You already saw an example of this. I just want to tell you a little bit more about what we're going to be seeing there, because each of these file formats is kind of complicated. It helps to understand a little bit what it is. So the name FASTQ comes from another format called a FASTA. And if you've ever looked at sequence data, like a sequence genome that you've downloaded from a database, it is almost always in the FASTA format. It's a very common format. FASTQ is the name comes from FASTA with quality, and we'll talk a little bit more about what that means in a second. One of my favorite web pages on the internet is the Wikipedia page for the FASTQ format. I reference it a fair amount because it's pretty useful. We're not going to look at it now, but if you want some good reading during a break while you're eating your lunch, trying to go to bed, I recommend it. So we're going to look really quick at an example of the FASTQ format, and also for comparison the FASTA format, because some people might be a little bit more familiar with it. So here is just a snippet of a FASTA format file. It is the complete genome for a specific strain W3110 of E. coli. And then this is a snippet from a FASTQ file, not related at all, but just for comparison. So there's actually a lot of similarities between the FASTA format and the FASTQ format. So I'm going to start with those, and then we'll kind of touch on differences. So they both have what's called a header. For a FASTA file, usually that's going to like be sort of sensical in the meaning that like you can there's not a requirement that it be sensical, but it's pretty common. Like I can look at this and tell, okay, well, this is from the E. coli, that's the organism. There's the strain is K12, the substrain is W3110, and this is from the complete genome sequence, right? So FASTQ files also have headers. These are almost always automatically generated by a machine, and so there's some sense to them, but it's it's less clear, and it's also they're made without direct human intervention. So the format for both of these, the requirement is just for FASTA that it just has this greater than sign at the beginning of the header line, and for FASTQ that it has the at symbol at the beginning of the line, and then everything else is sort of convention, but it's not actually a required part of the format. So for the FASTQ file, different sequencing companies actually have sort of a different default format. This is an Illumina sequencing file, and so there's actually a lot of information that you can get out of this file if you know how to read it. So this first thing here is actually the serial number of the sequencer that the sequencing was done on. This number here is the count of the number of times that this sequencing run was on the machine. So this is actually data that was generated from the 36th time that this sequencer was run. This is the serial number of the flow cell. I can never remember what this one is for, so let's ignore that. This is the coordinates tells you the coordinates on the flow cell where this specific read came from. So if you were here last week or 2 weeks ago, I guess last week when I talked about high throughput sequencing, I talked about how like the different spots on the flow cell are each going to generate a different sequence read, and that when the data is generated, that when the image is processed, the software keeps track of where on the flow cell a given base came from, so that you can string the sequence together. And this is actually what it's tracking. It's tracking the coordinates, and it's 3 sets of coordinates because this is the X and Y. And I just sort of touched on last week that they're actually sort of different tiles, different quadrants of the flow cell, that like each quadrant is imaged like with one picture. And so this is telling you the quadrant. And then this is some information that I'm trying to remember. The N means that this can either be an N or a Y, and I always find it confusing the way they did this. The N a Y would mean yes, this fails, our sort of standard quality control. So an N is good, and N means no, this doesn't fail. I would have done it the other way, but that's the way they did it. I think that the 1 here is telling you that this is the forward read, or the R1 from now. That doesn't mean that that's paired end, but it just tells you if it's paired end, there would be another matched read that would have a 2 here. And I can't remember what these zeros are for, but they're not super important. And again, that format is Illumina specific, and other sequencers are going to have a different format, and it's not a requirement of the FASTQ format. The next part of both of these files is the actual sequence. And so, this is the genome sequence of E. coli. This is like one read worth of sequence, right? And I think this is 75 base pair or 100 base pair reads. I can't remember. But for both of them, the sequence is the second part of the form. So now we get into something that the FASTQ file has that the FASTA file doesn't. And I touched on this a little bit last week, which is the quality score. And so for each base in the sequence in the FASTQ format, it has a quality score that's assigned to it. And that quality score is the sequencer, the sequencing software making an estimation of how accurate it thinks this measurement is, right? Because remember that the sequencer it's just like a ruler or a scale. It's an instrument for measuring something, and there's always error in measurement. And so this C is not, it's never 100% guaranteed that it's a C. It could actually, in the real organism, it could be a different base. And so this A here is telling us how reliable, how much we should trust this C. There's a algorithm for going from these quality scores that are characters into a number. For Illumina sequencers, the best sort of quality you can get is a expected error of 1 in 10,000, and it gets worse than that, right? So it can only be that's the best quality sequence you can get is essentially 1 in 10,000. You can get lower, but on an Illumina sequencer, you can't get any higher. And so there's an algorithm for converting from an A or an H or G into a estimated probability of error. We're not going to talk about that. I have some links from the website to a worksheet that kind of goes through it. It's not that complicated, but it's just I don't have the time to go through it today. And then, so that's the major difference between FASTA and FASTQ. I already touched on this, but these symbols are different. Oh, actually, I should tell you about this third. Oops. I should just mention this third line. So in the the symbols are different. Like I said, greater than for FASTA indicates a header. At for FASTQ indicates a header. Then in a FASTQ, you have these sort of quartets of lines. I actually don't want to call it a line. It's quartets of text. So the first part is the header. The second part is the sequence. The fourth part is the quality scores. And there's this thing here, and the FASTQ format says it has to start with a plus. It can either have nothing after the plus, or it can repeat the header after the plus. Either of those is fine. It's pretty common to just have nothing, because why do you need to repeat the header? But some databases, like the database that's run by the sequence database that's run by the NCBI, part of the National Institutes of Health, if you download sequence from it, they will actually repeat the header here. I will just mention, because this is a common mistake that people make if they know a little bit about FASTQ files and they know a little bit about programming. They think, oh, well, I know how to parse a FASTQ file. I just need to look for the @ signs and the plus signs, and I'm good. You are not good if you do that. At and plus are actually valid symbols in the quality score. And so if you just look for @ and plus, you're very likely to actually parse the file incorrectly, because you will find at and plus in the quality scores. You need to be, not going to get into details about what exactly you need to do, but you need to be a little bit more thoughtful about parsing FASTQ files. I never write FASTQ parsers because there are existing ones that you can use for all the programming languages that I use. The other thing that I will mention that people get confused about is that you can see here in the FASTA file, it's pretty common for the lines to be cut at 80 characters. And that's not a requirement. That is often done just so that it doesn't run off the edge of the page when you're viewing it. And it is also pretty common for FASTQ files not to cut the lines. So it's pretty common that you'll have the sequence line just keep on going. That, like I said, this is 75 or 100 base pairs. You could have a 200 base pair sequence read, and it's pretty common that it'll just keep on going to the right and not be cut. And the same for the quality score. But there is the option within the FASTQ format to actually truncate the lines and wrap them, just like the FASTA format. And so people assume that sometimes people assume that FASTQ format files will not be wrapped onto multiple lines, and that is not a correct assumption. Again, not something you need to worry about unless you are actually writing code to try and parse a FASTQ file. So like I said, what I just showed you before was just one entry in a FASTQ file. Often, you'll have files with millions to billions of those quartets, and you can see, this is a slightly larger snippet from a real FASTQ file, and you see, all the lines. It goes off the top and goes off the bottom, but you can always see, okay, this is a quartet because there's an at sign here after, coming after a quality score. So at sign, header, sequence, plus sign, quality scores. Any questions about the FASTQ format before I keep going?
Hi, Dr. Granick. I have a question.
Sure.
Yeah, so I'm just curious how the quality score of each base call is determined. Like, is it specific to a specific sequencer, or it's based on the intensity of the fluorescence in the flow cell, or like how the machine could know how confident you should, like how much you should trust this base call the machine is giving you?
Yeah, so it's a good question. So as far as I know, that is proprietary for the different sequencers. I've never really looked too hard, but I've never seen an explanation, like a detailed. I've never seen a publication of the algorithm for Illumina, at least. But yeah, so it's going to depend on some characteristics of the sequencers. So like for Illumina, if you were here last week, I talked about how you can have clusters that are really close together, and so you can't necessarily resolve the spots. So that's going to be sort of one thing that you can imagine would contribute to the quality score. Like you mentioned, it could be the fluorescent intensity. If there's just not a very strong signal, that's gonna reduce the quality. So, and again, we talked about last week how every cycle you're sequencing one base, and within that cluster, again, this is thinking about Illumina. Within that cluster, you have millions of fragments. Well, of course, all of those measurements are error prone, right? So you're getting an ensemble measurement of that cluster where it's let's say the cluster is mostly red, because that's in the fluorescence when you image it, because it's I'm trying to think. I think red is normally a T, but you're guaranteed, because both the measurement is error prone and the polymerase that's actually adding the base is error prone. You're going to get some, even if the correct base is T, you're going to get some misincorporation within that cluster because it's millions of molecules of other bases. And so that, even though like we look with our eyes and we see like, okay, well, there's nothing, I showed you, if you remember, I showed you the figure last week and you're like, oh, well, that's really red. You're going to get some other colors in that, from that spot. And so that's, also almost certainly going to contribute to the information. For the other sequencers, nanopore, for example, you're looking at current, and then actually using some deep learning algorithms to take that current information and then map it, figure out what base it is, and that's imperfect too. That's actually much more sophisticated, going from the raw measurement to a base call. And so I think that Nanopore, the algorithm, might be publicly available. So I haven't looked in, but you can kind of guess where, well, although it's, like I said, it's a deep learning model. So it's sort of, those are all a little bit black box. So you can kind of imagine where some of the quality information, like how you're going to go from the raw data to the quality information. But again, like I don't know, certainly not for Illumina, I don't know the details of how they make that determination. And yeah, so
Well,
maybe,
yeah, thank you.
maybe not a very satisfying answer. But there certainly for Illumina, you can think about how like the algorithms that are used for Sanger sequence data, those are publicly available, and you can kind of look and think about, okay, well, how does this translate to Illumina data? Okay, so just to give you a feel for, we'll take a break in a minute. Let me finish talking about FASTQ files. So we talked last week about paired-end data. And so these are like snippets. Actually, we talked last week about paired-end data in barcode files. And so these are snippets from the R1 or forward read file, R2, which is the reverse read file, and the I1, which is barcode file for a complete set of data. And as these sequences come off the machine for, I'll just say that this is, specific for Illumina data here. The sequences are in the same order that the sequences in each of these three files are in the same order, right? So this first read here in the R1 file corresponds to this. That is the reverse read in the, the forward read in the pair that goes with this reverse read in the R2 file, and actually this barcode sequence in the I1 file. And you can actually see that if you look at the coordinates of these spots, they're all the same for those three. And we can also look at each of these, I'm giving you a snippet with 2 reads in it from the file, just the top 2 reads in the file. And so you can see for this second read, the coordinates are all the same, right? So they're paired up. So if you've taken the file directly off the sequencer, they should be in the same order. Now, if you're taking the file and it's been processed in some way, it is possible that that order has been messed up and you can't trust it. So the way, if you really want to, you can actually check the coordinates, again, assuming this is Illumina data, check the coordinates and see that they actually match up. And some software will do that. Not all software will do that. And so we're going to get into, in a little bit, how this can create problems for you. The other thing that I'll point out is just that this other part, remember, this is the serial number for the sequencer. This is the run count, and this is the flow cell serial number. And so those are the same should be the same for all the reads in a given file, because generally you're going to have all the data from one sample from one sequencing run in one file. Now might be, if there aren't any questions about that, now might be a good time to take a break. Any questions before we take a quick break? So just to introduce you to the FASTQ file. So the first step in that's the input, both the input and the output for the first step, which is pre-processing quality control. So pre-processing, here's a sort of quick summary of what the pre-processing is. Demultiplexing, I talked a little bit about that last week. That is often done at the sequencing. These days, it's often done at the sequencing facility for you. We're not going to go through that today. I talked about it briefly last week about it's just taking the raw file and it's sort of dividing it up by samples using the barcodes. We'll talk a little bit about that. We'll touch on that a little bit more next week. Next week, because it's slightly more complicated with single cell data, but it's really conceptually very similar. The next step is quality control. Basically looking at this is looking at the data and like kind of trying to get a feel for like whether it's total garbage or whether it's okay or somewhere in between. We use that information in the next two steps, usually. We're going to do quality control hands-on when we get to that in a little bit. So I feel like it's a little bit more helpful to kind of actually be working with it in quality control, but just, that sort of summary information, not having, it's fairly general for all high throughput sequencing data. There can be some things that are a little bit specific for RNA-seq that you expect different than, say, DNA-seq. That's fairly generic. And then, like I said, that does sometimes inform our trimming and filtering. Trimming is, so I'm going to talk about that next slide. Trimming is basically removing parts of reads because there's some problem with the parts of reads. And then filtering is actually throwing out whole reads altogether. So they're similar, but slightly different. Just to talk about one reason why you might trim a read is based on the quality score, or we just looked at that. So if the quality score is not, you saw this a little bit already here, right? You can look at the quality scores here, and even without knowing exactly the mapping between quality and these characters, you can tell that it's not, consistent across the read, right? We see a bunch of H's here. G and H are, there is an ordering here, right? So G, let me think. H is higher quality than G. And so, and then F is lower quality than G, E is lower quality than F, there's an ordering here. It is actually like fairly consistent across here, right? There's this one that has a B, right? But all the scores are between B and H. So that's like what, 5 there's a log transform that goes into it, but these are all like somewhere all above 1 in 1,000 error rates, basically. But you can see that the quality is not consistent across a read. And so if you have you particularly tend to, at this end, at the 3' end, you tend to, it's more likely to have lower quality scores. So sometimes you'll trim off really low quality bases. Oops, my animation here. And then you might, if you just have a read that's total garbage, you might throw it out. The other reason why, there's a few other reasons why you might want to trim reads, but another major reason is what's called adapter contamination. We didn't touch on this before, but you remember I use this representation a lot. The orange and the green here are the adapters, and they're ligated onto the or attached to the biological sequence that we're actually interested in. So the gray here is the biological sequence we're actually interested in. And so you can imagine, let's say here, we're doing shotgun sequencing, and let's say that the biological sequence that we happen at, we fragmented this and we added on adapter. And so we ended up with a biological sequence of 230 base pairs with adapter on either end. So if our read length is 50, 100, or 150 base pairs, all we're going to be getting is biological sequence. But if we did a 250 base pair read on this 230 base pair biological sequence, you're going to get 230 bases of biological sequence and then 20 bases of adapter. Now, as far as the sequencer is concerned, when it's actually sequencing, it can't tell the difference between, there's no like chemical difference between the adapter sequence and the biological sequence, right? And the sequencer is just merely going to go along, and when it gets to the end of the biological sequence, it's just going to sequence next base, next base, next base, next base. And so the data that you get off the machine is going to be, from this fragment, is going to be 230 bases of biological sequence and 20 bases of adapter sequence. Now, of course, we already saw with the library example, when you're searching the database, it's going to be that you're going to be taking the sequence and searching the database. And this non-biological sequence, this adapter sequence that's going to come off the machine as part of this, could create problems in that process. And so often we'll want to remove the adapter. The way that we're going to remove the adapter with the data that we have is actually we're going to be searching the sequences for the adapter. We know the adapter sequence, right? That's the whole reason why we have the adapter on there. So when we're searching the sequence for the adapter, there's actually a nicer method that I like to use for paired end data. But the data that we're working with today is single end data. So we're just gonna be looking for the adapter sequence and trimming it off. Now, of course, you have to, you have to have some sort of threshold, because if you allow to trim any sequence that matches the adapter, with a short number, like if you allow a single base that matches the adapter, you're going to trim it. Well, every single base in the biological sequence matches somewhere in the adapter, right? The single bases are A's, T's, C's, and G's, right? So there's some threshold, and usually you want to kind of like say, well, it's got to match at least 8 or 10 bases of the adapter before I start trimming. So that you don't accidentally trim biological sequence that just happens to match the adapter. So that's the trimming is, and I'm going to walk through an example here right now. Here's some pretty common criteria that you use for trimming, and you do the trimming step kind of all at once, right? So what we're going to do is we're going to say if the quality score, so the name for the quality score is called Fred score. So if the quality score is less than 20, but 20 is 1 in 100 expected error rate. So, that's not super high quality. So that's not like a super stringent threshold. So we're using these characteristics. So we're going to trim anything that's less than 20, Fred score less than 20. We're going to trim any ends. So an N is where the sequencer has no idea what the base is. And so it just puts an N instead of putting an A, T, C, or G. And then we're going to be looking for this adapter sequence. And so we're going to search, we just have a little bit of our toy data here. We're going to be looking through the sequences for all these characteristics. So we're going to go through it step by step, but technically the program is doing it one by one, but it happens so fast that it essentially seems like it's doing it simultaneously. So the first thing we're going to look for is low quality reads. And so if you look here, this 2, that's a valid quality score. I don't remember what quality it represents, but I can tell you that, because I made this example, that a 2 is a Fred, represents a Fred score less than 20. And so usually when you're trimming based on quality, if you find, or adapter and the other problems, if you find a low quality score, often you'll search from the 3' end. Sometimes you'll also do a separate search from the 5' end. But if you're going to trim anything that like fails your criteria, you're going to trim there, and you're going to trim everything 3'. And the reason for that is that if we just cut out this base and cut out this A that corresponds to this low quality score. So if you just cut out this and then shifted everything over to the left, we're essentially introducing what you'd consider as a frame shift mutation, or a deletion mutation, a single base deletion mutation. Right? That's not the sequence that we observed if we take out this A and shift everything over. So it's considered more reasonable just to take out this base and everything 3' of it. So that is what we're going to do. With that base was, this A was low quality. We don't trust it. So we're going to just get rid of it and everything 3' of it. Okay. So next we're going to look for ends. You can imagine, I don't think I have any ends here, but you can imagine, you see an N, you're going to delete the N and throw out everything 3' of it. And I should mention that these values, I think, are the default for, I don't remember if it's the default for the software that we're using or not. I think maybe it is. They're fairly reasonable values. Whether you need to do all this really kind of depends on the downstream analysis. Some software, for example, it can't tolerate ends. So if you don't trim ends, then the subsequent analysis steps they'll die on if they see an end. And other software, it doesn't mind. For RNA-seq, I'm actually not entirely sure. I've never done this analysis. I'm not really entirely sure for differential expression how much you need to do trimming. I think actually, if I remember correctly, recently found a paper where it said essentially you don't really need to trim for RNA-seq for differential expression. It doesn't really have added value. For some methods, for some applications of RNA-seq, for lots of applications of DNA-seq, you do need to do trimming. So it's common practice, but like I said, it's not clear, and I think there's some evidence to suggest that you actually don't need to do trimming. We're gonna do it anyway, just because I think it's helpful to understand it, and you'll see it a lot. So there aren't any ends, so we're going to move on to the adapter. And so we're going to be searching for this, but we're not going to require that we find the whole adapter. We're going to allow partial matches. And if you have sharp eyes, you will notice that there's a partial match here. That's 15 base pairs. So that's unlikely to be, biological sequence. We get to do the calculation, but it's very unlikely to be real biological sequence. And so we're going to trim that too. Questions about that before I keep going? Okay. So now, there's a question. How to handle the A with the low quality. Look, so we're going to, the standard way for any trimming is that you trim the base or bases that are problematic and anything 3' of it. So here's the base that's problematic, here's the base that's problematic. I'm highlighting the quality scores. So this is again, I don't remember what Fred score this corresponds to, but it's definitely less than 20. So that's this A here. So we're going to take out this base and everything 3' of it, because if we took out just some of the bases, we're computationally introducing a deletion. And that is very likely to introduce problems, right? So we don't want to like we're essentially making up sequence at that point if we delete parts of fragments. The question is the adapter sequence the same for every gene? The adapter sequence is the same. We don't have, at this point, we're not thinking at all about genes. We don't know where these sequences are coming from, but the adapter sequence is, there's a little bit of, I talked about this some last week. There's potentially some if we're doing multiplexing, which often we are, there's barcodes in the adapters, but other than that, the adapter sequence is the same for every single read that we're getting off the sequencer. Now, whether we're actually going to usually, so it depends on the fragments we put in, but it's actually, especially if we do short reads, like 50 base pair reads, we might not see any adapter sequence. So in the output data, but yeah, that's the whole point of the adapter, is that it's essentially constant, because we need it to be constant, because it the sequencer requires it, right? And so both the oligonucleotides on the flow cell that are there for hybridization and the primer that's put in for the sequencing cycle, we expect it, the sequencer is expecting a specific sequence. So that's why we know that the adapter is constant. Now, I will say that there are, like, for example, on Illumina, there are a few different major sort of sets of adapter sequences. And so, so there is a little bit of variation that's allowed, but there's sort of two major sort of sets of adapter sequences. And so if you know which of those two to look for, then you can trim based on adapter sequence. I don't want to get too much into details. So the question, why not just replace the low quality bases with N? The 3' sequence of low quality base may have high quality and still be aligned to the reference. So it is true that replacing bases with low quality bases with N is not a totally unreasonable strategy in some situations. But like I mentioned, there's some software that we would use in subsequent steps, not the software we're using today, but for other types of analysis that just can't handle ends. And so if you substituted ends, it would certainly be problematic for that. For what we're doing today, you could get away with substituting ends. I will say that if you look at the distribution of quality scores across a read, it does tend to go down as you move towards the 3' end. And so once you start seeing low quality bases, there's a good chance that subsequent bases are much more likely to be low quality. And the other thing is that, like I told you, the quality scores are an estimation of the software. And so even though you might have, like here, you have a 2, which is relatively low, and then you have a G that is relatively high, this G might not, it might overestimate the quality. So it's a good point. I've never seen any sort of analysis that looked at the sort of relative merits of substituting ends for low quality bases in for software that can handle it. I recognize the sort of impulse to like not throw away data that like might be valuable. I will say that you end up getting so much data. In most cases, you end up getting so much data that throwing out low quality sequences is a sort of reasonable thing to do in many cases, because sometimes it's better to have like the best data rather than more like adding in kind of lower quality data. For RNA-seq, like I said, the trimming it's not clear that and there's some evidence to suggest that it actually doesn't give you additional value. So in that case, like maybe you shouldn't even be trimming at all. But there are situations in which it might be reasonable to substitute an N for low quality bases. That is not what most software does. I can't speak to whether there might be software out there that will substitute N's for bases that are low quality. I'm not sure. Yeah. Other questions before I keep going? Okay, so that was trimming. And I told you that filtering is throwing out specific reads. And so the criteria for filtering are going to be different than trimming. Here are some common filtering parameters. And again, these are almost always going to be adjustable with filtering and trimming software. And usually you use the same software to do it. And you do it sort of, again, simultaneously with do filtering and trimming simultaneously for reasons which you'll see in a second. So like here's some, somewhat reasonable common filtering approaches is we're going to throw out any reads where the length is less than 40. If the reads are too short, then they're not necessarily useful for us for many applications because we can't find the right place in the genome for them, right? Imagine in the limit where you trim down to a single base. So you have an A and you're trying to match that in the genome. Well, it's going to match, in a genome of 1 million base pairs, it's going to match about 250,000 locations, right? So at some point, the sequence is long enough that you have a good chance of uniquely matching at the genome. But if it's too short, you're gonna, not only does it add to the computational complexity because when you find multiple places, some of the algorithms you're doing kind of a lot more work. And so it's reasonable to sort of trim these or filter out short reads. And these are going to be, as they come off the sequencer, the reads are often all the same length. And so this is going to be usually it's going to be after trimming, that the length would be below the threshold. We can also calculate the average Phred score, the average quality score across the whole read. And so if the average Phred score is low, then we might want to throw out the whole read instead of just trimming. And then if we have a whole bunch of N's, that could suggest that there was like really a problem with that read, that spot on the flow cell. And so I'm going to throw those out. So we're going to search through this data after we've done the trimming. And so the length, this is now shorter than 40 base pairs. And so we're going to filter out this read. Now, then like these other reads are fine. Now, I mentioned to you that as the sequence comes off the sequencer, the order of the reads in the R1 file and the R2 file, they're paired up, right? So so this read in the R1 file corresponds to this read in the R2 file. And, we can, again, confirm that by matching up the coordinates. If I filter out this read from the R1 file and don't do anything else, I've broken that assumption, right? Because now if I look at what has become the first read in the R1 file, this one, it does not match with the first read in the R2 file. And so usually when you're doing trimming and filtering, if you have paired in data, you want to do it simultaneously on the R1 and the R2 file, because usually what you want to do is if you're throwing out this read, you want to throw out its pair, because otherwise you break that synchrony. And again, some software will check to see if the read pairs are synchronized, and some software won't. And if you don't know, or if you're using software that's not checking, then this will create a problem. And actually, even if you have software that's checking, usually what it'll do is it'll check, and if it finds any read pairs that are not in sync, it'll just stop. It won't bother to try and match them up, in part because that's sort of more computationally adds a sort of computational load to the process. So again, if we're going to filter out this read, we will almost always filter out its pair in the R2 or vice versa. So now we're going to, even though there's nothing wrong with this sequence in and of itself, we're going to filter it out because it's breaking that synchrony. We're going to calculate average Phred scores. I'm going to tell you that all of the reads that are left are fine. And you can just eyeball here, and we don't have any ends. So we're not in a situation where there are more than 3 ends in the read. And so this is what we started off with. And then at the end of trimming and filtering, this is what we're going to be left with. Questions about that? Okay. So let's keep going. So that's trimming and filtering. And so let's talk about mapping. We've already seen mapping in our library example, right? That's just taking this text and finding where it matches in the database. And, I gave the example that we're actually not only going to find the book that it matches, but also the specific location in the book. And so that is essentially what we're doing with the RNA-seq data. We're going to use this algorithm called STAR. It's a very popular software for RNA-seq data. Now, STAR adds this is actually a method that's used for analyzing a lot of high throughput sequencing data, including DNA-seq data. We are using an RNA-seq specific method because there's some a little bit added complexity that makes it so that software that's not specific for RNA-seq data will not do as well as software that's specific for RNA-seq data. And I just want to show you the kind of 2 major special cases that STAR handles, that software that's really designed for DNA data, that's not specific for RNA data, isn't going to handle, and so it's just going to not perform as well. So one is the fact that in eukaryotes, in general, you have exons, and actually more importantly, you have introns. So we're looking at messenger RNA. So it's been spliced. So the introns are no longer so before we've collected the RNA and purified it, the RNA, or most of the RNA that we're working with, has been spliced to remove introns. So this is what so assuming this is the gene, how it looks in the genome, the purple are rectangles are the exons, and then this black in the middle here is going to be an intron, which is going to be spliced out. So this is what the RNA, messenger RNA that corresponds to this gene is going to look like, right? It's going to have this exon ligated directly onto this exon. And so if you try and map this fragment directly to the genome, the sort of worst case scenario is that it won't match because, especially if you use like a really simple algorithm, it's not going to match because there's this big chunk of text in the middle here, that's the intron, that's not in the read, right? And so software that's asking for a perfect match is just going to say, well, I can't find where this goes, right? I find that it partially matches this, and I find that it partially matches this, but I don't know where it goes, right? So software that's designed specifically for RNA-seq is going to recognize that you might have sort of gaps in the genome that are not going to be present in the read sequence, and it can essentially do this split mapping. Software like STAR is actually going to kind of pre-process the genome, or STAR and other other software for RNA-seq will sort of pre-process the genome, and so it will actually kind of try to, it generates essentially what's called a reference transcriptome from the reference genome using the genome annotation, so that it just makes this step easier, so that instead of trying to map the read directly to the genome, it's mapping to the transcriptome. And so if there's a known intron, it won't have a problem. It's actually pretty easy for it to handle this. But it also has a component where it's going to be able to identify unknown introns where there's a chunk that's in the genome that is missing from the read. The other thing that software like STAR needs to deal with is that you will often have a poly-A tail, the polyadenylation that's at the 3' end of messenger RNA coming from eukaryotes. And so you can find that in the RNA reads, RNA-seq reads that are coming off the sequencer. And again, if it looks like this, where it's, the blue is the biological sequences that's in the genome, and the red here, or brown, I don't know what color it is, reddish brown, is representing a poly-A tail. That's not encoded in the genome, right? That is biological sequence that's being added by polyadenylation polymerase, but it's not in the genome. So if you try and map this whole read to the genome, you're not going to be able to map this. And so STAR, again, is aware that, there is this sequence that might be in the read, in the sequence data, that's actually not going to be in the genome, and it handles that correctly also. Questions about any of that? And again, the algorithm that's used is, I'm not going to go into the details. It's somewhat more sophisticated than that sort of like simplistic searching that I mentioned. And being able to do this, we're not going into the algorithmic details just because there's not time. But it's not bad to, I think it's reasonable to just think of it doing a sort of simple search, how you would do it by hand. So the input to this mapping step is the trimmed and filtered FASTQ, and the output is a file and format called a SAM file or a BAM file. So SAM, I can never remember what it stands for. So SAM stands for sequence alignment map. BAM is, the B is for binary. It's a binary version of the SAM file. So binary, it just means that it's much simpler and faster for a computer to read it than a SAM file. But you can actually convert between a SAM and a BAM file like really easily. There's software for doing that, and it's not lossy. You don't lose any information. You can go back and forth between a SAM and a BAM 20 times, and the file that you started with will have the same information as the file you end up with. It's just a slightly different format. BAM files, in addition to being much faster for a computer to read, are actually almost always take a lot less storage space than SAM files. For reasons that escape me, some software will only deal with SAM files. I don't know why, but most software these days, most software actually can work with SAM or BAM files. And I almost always, I always want to work with BAM files directly if I can, just because it's computationally, it's less simple. So this is the information that I'm going to use SAM and BAM interchangeably because essentially the information is the same. It's just on how it's stored is gonna, it's text. A SAM file is text. A BAM file is like, sort of incomprehensible. If you try and look directly at the file without using a conversion software, it's sort of incomprehensible. But the information contained in it is the same. It actually stores all the information that's in the FASTQ file. So it actually stores the header, it stores the sequence, and it stores the quality scores. But in addition to that, so you'll see that here, sequence, quality scores, I think name is header. That in the SAM file, they call it a name. But it is carrying mapping information. So it carries the information on the position in the genome where it found a match. It also can assign a quality of mapping, which is like, there's some specification as to how to determine that, but it's basically like how, again, instead, sort of like the Fred score is like how reliable the base call is. This quality of mapping is how reliable the mapping is. Most of the algorithm star that we're using, it basically tells you essentially how unique that mapping is in the genome. So if there are multiple places in the genome that kind of read maps equally well, the quality of mapping is going to be really low because there's uncertainty in whether a given spot is correct. If there's one place where it maps really well, but there's another place that maps like almost as good, that's going to be lower quality than if there's like one unique place in the genome that it maps. Within the SAM file, it also has information on whether there are insertions or deletions of the read relative to the genome. Like I said, it's got a sequence quality. If you have paired in data, it will calculate, most software will map as pairs, and it will actually calculate the size of the insert, the biological sequence, based on the locations that it actually mapped in the genome. There's some other information that's in there, but those are the things that we're interested in. I think I'm gonna not go through this. This is kind of, I could talk for a couple of minutes about how you kind of interpret a BAM file, but just in the interest of time, I think I'm gonna skip over that because I'm going a lot slower than I had planned. But this is, I'll just show you, this is kind of a toy representation of a BAM, SAM or BAM file. So this is the chromosome that it's mapping to. This is the, actually, sorry, no, this is some like bits of information about, we won't get into it. Oh no, wait, sorry. This is the header. I said this right. This is the header. This is the name of the chromosome here. The chromosome is just called ref. I believe this is the position in the chromosome and it's mapping. This is the mapping quality. This is what's called the cigar string, which is, I told you it's got that information on insertions and deletions. Not going to get into it. And then this is the sequence of the read itself. And it's not showing here, but like I said, it also actually has a quality string in there. And then this is some additional information that the mapping software like STAR is going to add in. You will see, you can have what's called an unmapped BAM file that some software likes using, because you can put information in a BAM file that there's this area here, this field here that you kind of like can add the format, the SAM BAM format allows for, creativity, some flexibility in what information is put here. So you can represent a lot of information in a SAM file that you can't represent in a FASTQ file. So for example, both Nanopore and PacBio will output BAM files that are unmapped BAM files. So they're not going to have the chromosome or the position information or the mapping quality. But the reason why they do that is because it has all the information that the FASTQ file has, and they can add in some additional information that is useful for some analyses. So it can be a little bit confusing because originally BAM files, SAM BAM files were designed for carrying mapping location information, but because has this sort of flexibility, its use has been sort of expanded. Why are some bits capitalized and others not on the BAM file? So good question. That is I don't think you would actually see that in a real BAM. So this up top here is actually not the BAM file. And this is sort of a toy representation. It's not exactly, it's missing some stuff. But here the lowercase bases are showing you where this is a read, and it's showing you where those lowercase bases are positions that actually don't match the reference. So it's kind of to indicate that. I don't think that that SAM files actually care about case. I'd have to double check the specification, but I don't think they care about case, and usually they'll just show you sequence in uppercase. I think that's right. Maybe not. Actually, maybe it might show lowercase for what's called soft clipping, where again it's like matching this and not matching that. I can't remember. I'd have to check. But yeah, if it does show that in a real BAM file, it's going to be bases in the read that don't actually match the reference. Other questions? Okay, so let's talk about counting. Counting is actually really easy, and it's often generally like star is going to do it. You can turn it off, but by default, it's going to do the counting simultaneously to doing the mapping. And it's just this step of like going through and then like sort of grouping the reads by here by book. The analogy for RNA-seq data, for the RNA-seq data, books roughly correspond to genes, right? So here we said, okay, well, all these reads are from a tale of two cities. For RNA-seq, we're going to say all these reads are, mapped to this gene. I'm not going to get too much into detail of this, but just you should keep in the back of your head. You don't need to worry too much about it, but in the back of your head, you can think about the fact that this is kind of the ideal case where a read is, here's the gene, and the read is landing somewhere inside the gene. There are different sort of pathological cases that are not what you'd expect, where a read is like partially on a gene, partially not. Where a read is actually, the read is spanning includes the intron. It's not exactly what you'd expect for normal splicing, but you see that sometimes. There are cases of overlapping genes. In this case, this read, you can unambiguously say, that read goes with gene A, but how do you assign this read? Does it go with gene A or gene B or either? And then how do you assign this read where it's like totally in the overlap? There are sort of different ways of handling this. We're not going to get into details, but it's useful to keep this in mind because every once in a while, this will show up and it will be an important problem. I had a data set where a bunch of reads were getting thrown away because they were like this. And so there was like no data from a given gene that collaborators said like, oh, well, we expect this gene to be expressed at a really high level. So it's helpful notes there. Most of the time you don't have to worry. But every once in a while you do. So the output format for the count tables is it's not bioinformatics specific. Usually it's tab separated values. It can be comma separated value, where basically you have kind of two columns, the name of the gene, and then the number of reads that were observed mapping to that gene. Pretty simple. Just so you know, we will see this. I'm not going to get, I think I'm going to gloss over this just again, because I'm running later than I would like to. There's some complications where we haven't talked about this at all, but there are some methods for preparing RNA-seq libraries where you can actually detect which strand of the genome the read came from. So for normal protein coding genes, you expect it to come from the one strand of the genome and not the other. If you've prepared the library in the way that you can, it's called the strand-specific library, you can then look and say, like, okay, are these reads actually mapping to the strand that I expect? So star will actually output 3 different columns where it tells you, let's see, so this is like the counts where where the data is like not strand specific. These are the counts where the reads are mapping to the coding strand, and these are the reads where the counts where the reads are mapping to the antisense strand. And which one you want to use depends on how the library was prepared. So some strand specific, most of the time, strand specific libraries will actually, you want to look at this column because it gives you the reverse complement of the RNA as it was in the cell. But you actually have to know how the library is prepared to know which column to use. And if you don't know that, there are ways to guess. There's software that can kind of give you some idea. But you also want to, we're not going to get into this, but there's also software that will, like, you can kind of use that as a quality control step, and you can detect problems if you know which column you should be looking at. And there's some problems with the data, you can detect them by knowing what the data should look like. So it's almost, you always want to, like, if you didn't prepare the libraries yourself, you want to check with who prepared the libraries and ask them, should I be looking for antisense strand reads? Should I be looking at coding strand reads, or should I be looking at the unstranded reads? I'm not going to get into too much detail. This is kind of showing you, well, I won't get into it, just in the interest of time. Questions about all that before we actually, now's probably a good time to take a break. So after I answer any questions, let's take a break, and then we'll actually switch over to the hands-on. Okay, while people are thinking of questions, I want to remind you, if you have not signed in, please sign in. I'm going to put the link in the chat, unless Richard just beat me to it. And then please, I will kind of quickly walk through the signing in. Actually, I'm probably going to sign in. Well, no, I guess I'll quickly walk through the signing in. But if you can go ahead and sign in to Open on Demand now during the break, that'll make things a little bit faster after the break. So question, does STAR need GTF, GFF annotation files to count? So the short answer is yes, you always want, if you're using STAR, you always want a GFF or a GTF. And I can't remember, I always like to give a GTF. I can't remember, I feel like it used to not be very happy with GFFs, but I could be wrong. But I don't like GFFs anyway, because they're, I don't know, they're kind of annoying. I think that you can actually run STAR without, without a GTF or a GFF. Just for people who don't know, the GTF and GFF are annotation files. So you need a reference genome to find where in the genome sequence the read is mapping. But let's say you have a human genome and you just have the genome sequence. So you're going to have 23 chromosomes plus the Y and the X. And so if you know that a sequence maps to chromosome number 4, and you have its location on chromosome number 4, but you don't know which gene that location corresponds to. All you have is the location information, and it's probably not going to help you answer biological questions. So the GTF, the annotation files, GTF or GFF, tell you, okay, gene W is on chromosome 4 from position 100,000 to 120,000. And actually, here's where the exon 1 is in gene W. Here's where exon 2 is, the start and stop of exon 1, start and stop of exon 2, start and stop of exon 3. And so almost always for RNA-seq data, it's going to be much more useful. And when I say much more useful, I mean like it's almost useless without the annotation. I can think of examples where you wouldn't need the annotation, but almost always you want the annotation. So hopefully that answers the question. Like I said, I'm pretty sure you can run star without an annotation file, but for differential expression, it's going to be essentially useless. Other questions? Okay, so let's take a 5-minute break. Okay, so I have logged into DCC On Demand with my my NetID and password. If there is anyone who is not here at this screen, or if you've went ahead and followed the instructions that I put in the chat and gotten into our studio, you're okay too. But if you haven't logged into Open On Demand, please let me know right now. Otherwise, I'm gonna push forward. Okay, and so I'm going to actually check my instructions so I remember how to do this. Actually, let me just point out, no, I'll get back to that in a second. Okay. I just want to start up the container with the right resources. Okay, so if you are here, I think that, let's see, I think I can click on here, the all available apps. You can also click on right here. Oh, no, that's confusing. I'm going to click there. I'm just going to get rid of those things. Okay. Okay. And so if you have not already opened RStudio, you should be here. You want to click on the CHSI RStudio workshop. I'm assuming everyone has done this before, so I'm going to go a little bit fast. But please yell if I'm going too fast for you. So we are going to do things pretty much, so we're going to change a few things. So we want, in order for the software to run faster, we want more than the default resources. So under CPU, you want to put 10. And under memory, you want to put 64. So we're asking for more resources. If you have, so under account, you want HIV R25 2024. For most of you, that will show up as the default, but some of you, if you have another account on the cluster, then it might not show up as that. And again, for most of you, it should default to CHSI high, but if it doesn't, you want to select CHSI high. Anyone, and then for singularity image, it should default to the R25 HIV AIDS workshop. If it doesn't, please select that from the menu. Anyone not have this? Okay, so now I'm going to click on launch. I'm just going to give that a minute to start up. Now, while I'm waiting for that, I'm going to check and make sure I cleaned up after the last time I ran this so that you're getting the same thing that I've getting. Come on, stop it. Okay. So now I'm going to click on anyone not here. So if you're having problems, if you can either put it in the chat or if you can directly message Richard, and I'll let him help so that I don't have to, everyone doesn't have to work through the problems. And if Richard can't troubleshoot, then someone can yell at me. Okay, so if I'm assuming almost everyone's here, so I'm going to click on the connect to RStudio server. And so you might start up looking. So what you want to look for, again, I'm assuming most of you have been here before, but you want to look and see what it says for project up here. So if it says none, that's fine. If it says, let's see, what would it say? Oh, I think I actually need to do, oh no, I'm good. If it says 2024, 2025 HIV workshop, you're good. So right now mine says none. So if yours says none, you want to click here on project and you want to find HIV 2024, 2025 HIV workshop. And I'm going to click on that. Does not exist. Oh, okay. So I am going to clone the repository. Does anyone not have either they're already in the 2024, 2025 HIV workshop already, or they've found it in the menu? If you don't see that, yeah, so don't, oh, okay, okay, fine. So a lot of people, a lot of you are new. Great. So we'll walk through this really quick. So I want you to go through, yeah, I guess that makes sense that a lot of you are new. I should realize that. Okay, so I'm going to put, you can use this QR code if you want. I'm also going to put the link in the chat. So this is the link to the all the workshop material. We're going to do what's called cloning the repo. I'm not going to get into what that is right now. I can, if people want to know, I can point you to some of the older videos that covers that. But I'm just going to walk you through what you need to do so that we can do a quick RNA-seq analysis. So you should go here to the link that I put in the chat, which should get you here. So the next thing you want to do is you want to click in the upper right. There's this blue button that says code. And if you click on that, it's going to pop down some different options. I think that you all should be able to see that, but if not, it's okay. I mean, sorry, you should be able to see that on the screen that I'm sharing. I think we'll see two different options, clone with SSH and clone with HTTPS. You want to do the clone with HTTPS. So that means clicking on the clipboard next to that. If you want, you can also like click on that to highlight it, but I like just clicking on the clipboard. That'll copy the URL to for you so that we can use that in a second. Anyone not get that copied? Actually, I'll put it in the chat what I just copied, but it's probably easier if you just copy it from the code book. Okay, so now we need to go back to, and I should have said this before, you either want to keep your RStudio window open. So if you don't have anyone not have their RStudio window, anyone close their RStudio window, and need me to show you how to get back to it. Okay, so if you go back to your RStudio window, this is not too hard. Hopefully, there's potential that there's a problem, but well, let's assume that it'll work for everyone. I think the defaults are set right. So click on project, and you want to select new project in the menu that pops up. And that should pop up a box that says create project. And there are three options: new directory, existing directory, and version control. You want to click on the version control. And again, if I'm going too fast, please yell, and I can do it again. Okay, so now you should have the option of Git or Subversion. We want Git, so click on that. And that'll get you here to this clone Git repository window. So in this first thing at the top, the repository URL, you want to paste the URL that we copied from the Git repo, and the last one, the most recent one that I put in the chat that ends in .git, same thing. So I'm going to paste that on a Mac. It's Command V. On a PC, a Windows computer, it should be Control V. And if you're on Linux, you probably don't need me to tell you what to do, but I can. Anyone not got the URL pasted in there? And you can't see the whole thing because it's long, but it should end in, 2024, 2025 HIV workshop.git. Anyone not there? Okay, great. So if your cursor is anywhere in there, and if you push on the Shift key or the Control key or the Option key or the Command key or I think even the Windows key will work. Maybe don't do the Windows key. Do Shift or Control. Then it will copy this last little bit here, and it's going to plug it into the project directory name. If you can type it or you can copy it from here, but it's just easiest if you just push Shift or Control or whatever. Anyone not get that? Great. And then the last thing is I'm going to actually change this. I'm going to recommend that everyone clone this into their home directory. That's not where I normally do it, which is why you're seeing other things here. But I'm going to show you, if you have a tilde there, you're all set. If you have something besides a tilde, I want you to click on Browse, and then click on the Home up here. And then it should show a tilde right there. And click on Choose. And so now you should have a tilde there. Anyone not have that? Okay, great. So I'm going to click on Create Project, and it's going to do some stuff for like a few seconds. And then, it's going to go back and forth here. No, it's actually taking more than a few seconds. Maybe it's going to take 15 seconds. Okay. And so now, once it's done, if you just did that, you should have now, it should look like this. Up in the top right, it should say 2024-2025 HIV workshop. This window should look the same as mine. This window, you might have a source pane up there, but otherwise it should all look the same. Anyone not there? Okay, and then for those of you who have been in the workshop before and you already had this here, you probably know this, but what you need to do is you need to click over to the Get pane up here and then click on Pull. So mine, because I just cloned it, it's going to say already up to date. Okay, so people are saying, I don't see his workshop in the menu. Same with me. So did you just go through all the steps that I went through to clone the repo? What does it say up there? Does it still say no project? Oh, uh, oh, sorry. Is there anyone who's not here with 20? Sorry. I see someone said that those are old questions. So is there anyone who's not here with 2024, 25 HIV workshop up here? If not, let me know now. Otherwise, we'll got to keep going. Yeah, so there's a question about not seeing all the steps. Yeah, let me put in the chat where the steps are. Okay, give me a second. So I'm going to put this in the actually here. I'll put the direct link into the chat, but I'll also show you really quick how to find it. I'm just going to do this for everyone. There. So if you go to the link that I just put in the chat, it gives you what I just walked through. Should have done that before, sorry. If you're here, just for future reference, if you're here and you want to get that information, it's linked from here, initial download of workshop content. Okay, so is anyone else not here? Fantastic. Okay, so I just want to show you what we're gonna just like a quick overview. If you click on this link here, the Bioinformatics workshop schedule, so these are this is today. This is the link to the slide I should have told you before. This is the slides that I went through earlier. And then we're going to do this R Markdown 1, then R Markdown 2, then R Markdown 3, then R Markdown 4. I have like a little side hidden summary of all these steps, and there's some bonus material. If you click on the table of contents link here, these are the same. This is R Markdown 1, R Markdown 2, R Markdown 3, R Markdown 4 that's in the schedule. This is the lecture slides. This is some bonus material. And this is the link for actually the details about how we open RStudio with the extra memory and extra CPUs. We are going to be using some like basic Unix commands. I'm not going to go into that. We just don't have time. But if you want to learn, so if you just follow what I show you, you'll be fine. If you want to learn more about Unix, here's an intro from Software Carpentry. And then this is here's the intro from Software Carpentry. If you want to run this in Open On Demand, you need to do this step, this setup for Software Carpentry Unix lessons to just download the files that are used in this example. Again, not going to go into that right now. Okay, so what we're going to go through is quality control, trimming and filtering, downloading and indexing a genome, and mapping reads to a reference genome. So we're going to open those files in RStudio, but if you want to look over them, you can also find them here. Okay. Any questions before I keep going? Cool. Okay, so if you are here, if your file browser happens to not show the same thing as me, as if it up here it shows 2024-2025 HIV workshop, you can click on this R in a cube there, and that'll get you here. So I'm assuming everyone's here. What you want to do is you want to click on the workshop 3 bioinformatics, then on lectures, and just in case you get lost, you can see where we are right here. So click on lectures, and then the files are numbered in order. So this is the first set of, sorry, this is day 1, what I did last week. So day 2 is today. So 01 is the lecture slides. 02 is the first or markdown file we're going to use. So I'm going to click on that. It's called 02 day 2 FastQC. And then I'm going to increase the font a little bit. Is that an okay size for everyone? I know that like different people have different size screens. So anyone want me to make it bigger? Okay, great. So we're just because of time, we're just gonna kind of walk through and run chunks. Normally I like to, when I have more time, I like to kind of type things out. But like I said, I'm going slow today, and so I'm just gonna try to quickly explain what's going on without getting into too much details. Okay, so you're going to want to run like each chunk, and if you don't run the chunks, if you miss one out, then you're going to get error messages later possibly. So the first chunk here, we're just loading a library called FS. It's a library that's helpful for, this is an R library. It's helpful for working with like file names and things. And we're going to be using that in a little bit. This next chunk we're going to load is telling, we're setting up some variables about where we're going to be finding files, the input files that are the FASTQ files, and also where we're going to output stuff. So this is a shared directory that has all of the, this is publicly available data, all the FASTQs we're going to be working with. Actually, I should give you a little bit of background, very quick. So this is HIV-related data. It's actually not from humans. Because there are all sorts of complications with working with human data. And this is just simpler. So this is data from a fungus called Cryptococcus neoformans. It is a opportunistic infection that is particularly a problem for people with suppressed immune systems. So it is in the United States, people who have AIDS. So where HIV has progressed to actually having like a strong immunosuppression. Cryptococcus neoformans infections is one type of Cryptococcus neoformans is one type of infection that you see in people with AIDS. In the United States, again, it's pretty common in the United States. It's rare in people with a normal immune system, normal healthy immune system. You see it in people with AIDS, people who are getting some sort of immunosuppression for some other medical treatment, so like organ transplant, chemotherapy for cancer. It can happen in people who have inborn immunodeficiencies. The place where it used to be observed before there were like sort of common diseases and medical treatments that were strongly immunosuppressive was actually in people who kept pigeons. It's Cryptococcus neoformans infection is also known as pigeon handler's disease, because in their guano you find pretty high levels of, or you can find pretty high levels of Cryptococcus neoformans. And so if you're exposed to lots and lots of it, you can get, even if you have a normal immune system, you can get infections with this fungus. Okay. So that's the shared data we're going to be working with. And then this chunk is actually making, when you're working with sequence data, the input files can be quite large, and at the intermediate steps, like the trimming and filtering, we're going to generate like fairly large files. So this is like a detail of the Duke compute cluster that we're working on, is that, and it's going to be, if you're working on different systems, it's going to be different. But there are a couple of directories that are used that are essentially working directories, or like scratch directories, where you can put data, or actually more importantly, you can put like sort of intermediate results, and they get stored there. It gets automatically cleaned up in this directory, this what's called the work directory, every 60 days. I actually like that because they clean up after me, and then I don't feel so bad being a slob. But it's important to note that if you are using Duke Compute Cluster for your own research, you should never put data in /work that is precious to you, that you can't replace, right? So you should always have, if it's raw data, like, our raw data is here. If you have raw data, you shouldn't have your only copied in /work, because it will get deleted in 60 days and then you will be sad. So /work is temporary storage. There are a couple other places on Duke Compute Cluster where you can store, there are places where you can store like long-term data, and there are places where you can store temporary data. We're going to use /work. This chunk of code is for everyone. It's going to make a subdirectory in /work that uses their net ID, and then a subdirectory of that called HIV 2024, and then a subdirectory of that called RNA-seq. And we're going to be outputting all our sort of intermediate results into that directory. And again, because it's using our net ID, everyone's net ID, which is the program, the code is grabbing here, each of us will have a separate output directory. So we're going to make, because this file, we're doing the QC step, we're going to make a QC output directory. This is grabbing information about how many, if you remember when we started up the RStudio, we asked for, I think, 10 CPUs. And so this is just getting that information from the system, that we have 10 CPUs available for us. So in subsequent steps, we're going to tell the software we actually want to use all 10 CPUs. And so that's what this step's doing. This is actually this next chunk. So I'm going to run that again. I think I ran it, but it should be good. It doesn't actually generate any output. I probably should change that. So this step is actually making that QC output directory. And then here, we're just going to check and be sure that it got made. We're asking for file info. If the directory wasn't made, there wouldn't be any output here. The fact that it says is dir, that's just saying, like, is this a directory? Because this could be a file. It says true. So we're good. If you see something, if you see this all blank, then you need to rerun this chunk here. Okay, and this is gonna be, I'm not gonna get into too many details here, but this is so far all of the code that we've used so far, and I think everything that you've done in the workshop series, if you've been in other workshop series before, is in R. Like I said, we're going to be directly using Unix commands. And so in order to use Unix commands and have variables in Unix, we need to do this. This is making a Unix variable called raw fastQs from this R variable called fastqdir that we defined up here. Somewhere here, fastqdir. And I really like organizing things into variables because it makes the code more reproducible. We don't have to go through in other places. If we define all this stuff at the top, we don't have to like dig through the code and find places that if we want to change things, change directories, stuff like that, we have to find. Okay, so I mentioned that we're going to be doing the quality control, and that sort of first steps of that, we're going to use a program called fastqc. If you have a Unix program and you want to find out how to use it, you can often, this is not always the case, but you can often run the program and then as a parameter, either say -h or --help, and it'll give you some information on how to run it. So you run this chunk, fastqc-h will give you some information on how to run it. I'm not going to go through all this. We're not going to use all these steps. I'll tell you or all these sort of options. I'll show you the ones we're going to use, but just so you know, this is actually helpful. Information. Okay. So I'm going to start this chunk running because it'll take maybe a minute to run. And then while I'm doing, I'm going to tell you about it. So this first thing here, set-u, it's not going to get into details, but it's a feature of Bash, which is the Unix shell that we're using, that it does a little bit of like sort of debugging for us. If we misspell, these are the variable names that we defined up here. If you remember this, way up here, we define raw fastqs, qcdir, and num CPUs. So to actually use those variables within Bash, we got to say $ and then the name of the variable. If we misspell this variable name, here with set-u, it would actually give us an error message saying, I don't know what that variable is. Without set-u, it would just go happily along. There's actually many programming languages would give you an error, but by default, Bash does not. So saying here, having set-u tells it, okay, give me an error message if I misspell things. So this command is actually already done. We ran fastqc. --threads is a way to tell fastqc, okay, actually, I want you to use all the CPUs that I requested. So that's this value that's in num CPUs. So it's 10. That's what we set when we ran it. --extract is actually going to tell it, normally it packages things, the results up in a zip file. You've probably all encountered zip files. We would actually look at the results. So --extract is going to tell, okay, don't package stuff out in a zip file, give us the raw file so we can look at it. We're just, for simplicity, we're just running on one fastq file. It's this one. It's just one of the samples. It's in the directory, the shared directory that we can all access that has the fastq files in it, the raw data. And then this is telling it, -o is telling it where to output the results. And again, this is a variable that has the path that we defined at the beginning. Okay. And so I'll make this smaller so we can see. So that we can see, take a quick look at the QC output. This chunk is going to tell us that directory. So you can either follow what I'm doing or just watch. If you copy this, highlight it, and then on a Mac, I'm going to do Command-C. On a PC, it should be Control-C. And then if you click on the 3 dots here, it'll pop up a little dialog box where you can paste with, again, on a Mac, Command-V. On a PC, it should be Control-V. You could also type it. A little long for me to type, and I like typing. And then if we click on OK, this is the output from fastqc. So I'm going to click on, we want this, this is the fastq file name, then we added on the fastqc. No, I guess the program did. And then it's .html. So there's a web page. And if you click on it, then it's going to pop up this dialog box, open an editor or view in web browser. I'm going to click on view in web browser. And for me, it's opening this as a new tab. So given that I'm going really slow, I'm going to go through this really quick. So this is the output from fastqc for one file. Oh, actually, you know what? Give me a second. I'm going to flip back over here. I'm going to run the next few steps because these will take a little bit longer to run. And then while those are running, I'm going to do that. So this next step here, this is going to actually run fastqc on, well, this is this is the files it's going to run on. It's going to run 10 different files, just because the next step after this, it's more helpful to have multiple files. So this chunk here, 83 to 85, that's going to run it on FastQC on all 10 of these files. And I'm going to actually run these next two chunks. They won't run, they won't run yet. They'll run after this chunk's done. So I'm going to run that one, and I'm going to run that one. Now I'm going to flip back over here, and we'll take a quick look at the output of FastQC. This stuff up here is kind of basic information, the GC content, the average length of the reads. If you want a summary, you can look at this. Anywhere where there's a green check mark, FastQC is happy with the sort of quality control information. If there's a yellow exclamation point, it's concerned. It doesn't mean that we should be concerned, because this can vary depending on the data. If you see a red, I think it's a red X, it's really concerned. But again, how we use that information is going to depend. So this is looking at the quality scores, lining up all the reads in that input file and looking at the average quality scores in each position. And what you can see, this line is the, I can't remember, I think it's the mean, could be the median, I think it's the mean quality score in that position. And you can see that like the first, what is that, 6, 5 base pairs, a little bit lower, the average quality score is between 31 and 32 Fred score. And then it goes up. That's pretty common with Illumina data, that the first few bases are a little bit lower. And then it goes up to about an average of 35. And then it's, like I mentioned, it's slowly going down. But even here, the average quality score is 30, which Fred score 30 is a 1 in 1,000 expected error rate. And then these are just like the error bars on the interquartile range on those Fred scores. So all the way out to position 76, the average is about 30. Here's just another way of looking at it. That if we take look at the whole read, and we talked before in terms of filtering, the average Fred score across the read, you see the average Fred score across the read is, for most of the reads, is 35. You have some where it's a little bit lower. So that's all pretty good. Here we're looking at the distribution of bases across all the reads. Now, this is what we want. We want it to see, so this is yellow is G, blue is C, and then green is A, and red is T. So we want them to be parallel to each other. The actual values here is going to depend on the GC content of the gene. So all of this is good. It's giving us an exclamation point because at the beginning here, they're not parallel. This is pretty common, and I've got all sorts of hand-waving explanations for it. I'm not going to get into it now. If it's 10, 20 bases, 10, 15 bases that are like kind of squiggly like this at the beginning, I'm guessing it's probably a function of how the library is made. It could also be a few different steps in the library prep. I'm not too concerned about that. This is looking at the average, looking across all the reads, the sort of distribution of the average GC content. It gives you, the blue is the theoretical distribution, the red is observed. It's pretty close. It's giving us an exclamation point because the theoretical doesn't match the observed. This is not that. This is almost certainly, I would guess this is actually probably because of this, which again is probably a function of the library prep. This is looking at the ends in all the positions across each read, like stacking up all the reads. There are some ends in there, I think, but they're almost undetectable. So we're pretty happy, and it's pretty happy. This is the sequence length distribution. All our reads before we do trimming are 76 base pairs long. Great. This is sequence duplication. So this is where you're seeing either whole reads or parts of, I guess sequence duplication is, I think, whole reads that are, we're seeing the same sequence repeatedly. For RNA-seq, this is not surprising. For DNA-seq, so you're seeing like some duplication. That's, for DNA-seq, this would be a little surprising because you expect, even representation across the genome. For RNA-seq, we don't expect that. That's the whole point. We're looking at differential expression. Some genes are expressed at higher levels than others. And so that's part of the reason why we can have these sort of like sequence duplication with RNA-seq. Usually I'm not concerned. This is looking at overrepresented sequences. So like really high levels of sort of repeating sequence. And it will tell you, it's got a database of some common overrepresented sequences. In this case, TrueSeq adapter, that's the adapter sequence we use. And so it's saying that we have 3,000 occurrences of sequence adapter. That's adapter contamination that we talked about before, right? So again, we're going to trim that. Not a big concern. I mean, this is, you get some adapter contamination. And this is just another way of looking at it. It's showing you like several different adapter sequences that could be in there. It can also detect poly A and poly G. But it's actually like pretty low level. So it's not, it's not even registering on this. Any questions about all that? Okay, I didn't go through as fast as I meant. And then, so I already told you what all this is doing. This is just, we're just running on multiple files. And the reason for doing that is, so I can show you multi-QC, which is, you might imagine that, so for this data set, I think there are over 40 sequence files from like 40 different samples. Trying to look at all individual FastQC reports from all those, it's like, mind-numbing. And this actually isn't like a super big project. Multi-QC provides summaries of FastQC output, actually provides summaries of other stuff. So again, here I'm running multi-QC-H just to give you some information on how you run multi-QC. Here I'm just running multi-QC. Here is this the directory where we put the FastQC output. And then so multi-QC knows how to read a FastQC output file and then sort of summarize it. And then the output, we're actually putting the output from multi-QC in the same place. That's where our QC information is. And then if you run this chunk, it'll tell you, again, is actually the same place, but you can copy this, click on the 3 dots, paste it here. And now it's opening here. And so now we want to look at the multi-QC report. So if I click on that, view in web browser. So this is a summary of those files that we just looked at. So I'm not going to go through it again because I'm really running late, but you can see now it's showing you multiple files. Okay, so that is it for the QC. I want to now click on the R in the cube. And we're going to go back and find the next R Markdown file. So R in the cube should take you here. Workshop 3 Bioinformatics. Lectures. And then next we want to do the fastq trimming.rmd. So click on that. Any questions? And unfortunately, I don't think I'm going to be able to get through everything, but in 5 minutes, I'm going to call it a day so that there's time for everyone to fill the survey. But the materials here, if you want to walk through it yourself. Okay, so here we're doing FastQ trimming. Where I already talked about that in lecture. We're going to load a few libraries that we're going to use later in this chunk. We're doing things a little bit differently here. So there's actually a config file that is somewhere here in this miscellaneous directory. that has a bunch of these in the FastQC file. I defined at the top these paths. It's actually, I find it really helpful for like the projects like this where I have multiple R Markdown files that are doing different steps, to actually take this sort of information and put it in one centralized file. So I can show you it really quick. So here you can see I've defined a bunch of different paths for the different steps. And so it actually makes it a lot easier to, for example, if I want to move this project to a different cluster, I only really need to change, I think, these top two things here. And I don't have to go into each of those files and move it. And so this is how I tend to do work. What this step is doing is it's using source, which is an R function, to load that config file. And so all of that, and actually, if you have your environment tab open, you'll see when you run that chunk, you get the environment gets populated with a bunch of variables. Oh, num CPUs is not working. Oh well. Okay. So then there's this directory trimdir that we define, well, that I defined in the config.r file. This variable. That's where our output from this step is going to go. And here we're just making that directory for output. Then we're doing the same thing we did with the QC directory here. We're doing file info just to confirm that that got made. And you can see directory type is directory, or the type is directory. So that's good. So we're going to use a program called Trim-O-Matic for this, for the trimming. Actually, like I mentioned, I have another way that I like to do it, a program called FastP that I like to use for paired end data, but this is single end data. So we're using Trim-O-Matic. Trim-O-Matic actually has like two different versions. There's Trim-O-Matic SE for single end data and Trim-O-Matic PE for paired end data. For Trim-O-Matic, if you want to see how to run it, you can actually just get a summary by running the Trim-O-Matic command without any arguments, and it gives you this somewhat not very helpful summary. There's a Trim-O-Matic's like a little bit old, which is one of the reasons why I don't like it so much. There's manuals that are available online for it that give more information. In order to use Trim-O-Matic, we actually have to make an adapter file. I've already done that. That's in the Git repo, the Git repo. This is what the adapter file looks like. So it's a FASTA format file. Here, this chunk of code is just actually demonstrating showing you the contents of the file. So this is the name for the sequence. Names aren't super creative. Adapter, read to adapter, or adapter read to. The reverse complement of adapter, and then the reverse complement of adapter read to. And these are the adapter sequences here. So we're going to feed those into, well, actually here, we're going to do a little quick demo here where we're going to use a Unix program called zgrep and use it to use this file to do a quick search through the FASTQ file, one of the FASTQ files. And then this is doing a count. And so basically it's finding 87 instances of one of these. We're not getting into which one, just to keep it simple. So it's doing a count of how many times it finds this full length adapter in this file, right? And remember, before when we did trimming, we didn't require the full length sequence. So this is sort of extreme cases that it's finding 87 instances of full length adapter. And actually, if you run this chunk, you can, wait. No, sorry. This gives you a command. Here, I'll do it. You can run it in the terminal and actually see here where you see red, it's coloring in red all the adapter sequences in all of these 87 reads where it had a full length adapter match. So Trimatics can be a little bit more sophisticated. It's not going to require a full match, and so it's going to find more. I'm going to start this chunk running. I will explain it really quick, and then I think we have to call it a day because I was slow. So here, Trimatic, here's a Trimatic command. Threads is like before, it's telling Trimatic, okay, yeah, we actually have several CPUs you can use. So this is the number of CPUs you can use. This dash Fred 33, I didn't get into this, but for the way that Fred scores, that quality scores are represented, there's sort of two major categories. There's Fred 33 and Fred 64. And almost all the data that you get, pretty much if you get data off a sequencer today, as far as I know, it's going to be in the Fred 33 format. There's a period of time where Illumina was generating Fred 64 data. So if you have old data, it could be Fred 64. Again, if you have data that was generated really in the past 5 years, you're probably safe in assuming it's Fred 33. But so this is telling Chromatic that this is actually Fred 33 format data. It's an unfortunate feature of FASTQ files that there's no perfect way to tell from the FASTQ file whether it's Fred 33 or Fred 64. There's a couple of ways that you can kind of get a pretty good guess, but it helps if you know. Actually, I can tell you. So here, this in the FastQC output, this helps tell you. But again, this is actually estimating from the data. It's not. It can be imperfect. This is telling it. This is the directory to find the input FASTQ file. Sorry, this is the name of the input FASTQ file. And this is what we want to call the trimmed FASTQ file. And then this is telling Trimmatic exactly how to do the trimming. This is telling it where the adapter sequence is. I'm not going to get into all this stuff, but basically it's saying if the read is 36 base pairs or shorter after trimming, throw it out. It does kind of window where it moves along and calculates the average Fred score within the window and will trim. I think it's, I can't remember if it's the 30. In the notes, I explain what all these values are, and I can't remember off the top of my head. But these are all the trimming parameters that we already talked about. And then this is the sort of kind of summary of the output, and it's telling you that these are the sequences it used for trimming. This is the number of input sequences. This is the number after trimming and filtering. So 98.4% of them survived the trimming and filtering step, and then 1.6% of them were filtered out, thrown out. I like to do the rest of this is actually doing just a little bit of a kind of reality check on this. This is the trimmed output file. And then this is taking that output file and doing that same sort of quick and dirty, looking for the full adapter length in the output. And we see that now there aren't any. We don't see any adapter left. So it seems like the filtering worked. The trimming and filtering worked. So we need to call it a day because we are out of time. I'm sorry, like I said, that I didn't get as far as I planned. Just moving slow this morning. If you want to keep walking through, like I said, R-markdown 3 and R-markdown 4 here are the next two steps. I didn't talk about indexing a genome, but that's you need a reference genome to map to, and indexing is this sort of pre-processing step that STAR needs in order to be able to run quickly on the genome. That's what happened in this step. And then this is that mapping step to a reference genome. And then that's going to generate those tables that I showed you a representation of. And that's what you're going to use as input in 2 weeks when Dr. Shi walks through the the statistical analysis. I need to give you, actually, Richard, do you have the link for the survey? Actually, I'm going to put in, if you did not sign in, say this one more time. If you did not sign in, here's the sign in. And Richard just put in the link for the survey. Please fill that out. We need your feedback, not only to try and improve things for next year, but also to report back to our funding agency. Yeah, so please take 5 minutes just to fill that out. And that is how you can thank us for providing these workshops for free. Any questions? And actually, I will just one more time, I'm going to put in the link where you can find this in the chat. Oops, I messed that up. Do that again. Okay. No questions? Everyone good?
Hi, Dr. Granick.
Yeah.
Yeah, I did see the email you sent earlier about the afternoon session today. So it's the amplicon
Yep.
sequencing one, right? Okay, so I was talking to another classmate in CE 690. I think we're just a little bit confused, like how like after today, how the future sessions are going to be held, because we got emails from Kelly to RSVP the like this session, but we just now realized that there are like two different things. So do you have the entire schedule for like this class after this week?
Yep, and I'll email you, and I gotta think about where this schedule is. But I'll email you, and if you can sign up, there's a link for signing up. That would be helpful. But I can also check in with Kim and make sure she adds you to the email list. But I'll send that to you in the next I'll do that now. I just have to do it.
Okay, okay. Thank you so much.
Okay. And there's a question that I think someone asked about copying the repository into your local RStudio. And the answer is yes, you can do exactly the same. You can do exactly the same steps that's here. And I'll put the link in the chat. But your RStudio almost certainly does not have all the software installed that we use today. So if you try and run the code that we use today in your local copy of RStudio, it's going to almost certainly choke. And trying to install it is kind of complicated. We do have a container that is publicly available. I can put the link for that in the chat, but trying to run that on your local computer is, it requires, so there are two reasons why we actually do all of this work in Open On Demand. One is that it has the computational resources, both memory and CPUs, to do this, but also so that everyone's working in the same environment. And so part of that, oops, oh, I clicked on the wrong thing. And so part of that is that we have this container that it's really easy to run on Open On Demand, and it's a little bit more complicated to run it on your local computer. So I'm going to put this in the chat. Again, this is, a little bit more advanced than the sort of level that we're talking about. But if you want to try, you can, there's some not so good instructions for downloading the container image and running it. You need to install Aptainer, which I'm not going to get into right now. Any other questions? Okay, if you have not already filled out the survey, please do that now. Someone had a question, Docker not working. So this is actually a Singularity container or Aptainer container. In theory, you could use Docker. I think there is a way to convert an Aptainer or Singularity container to Docker, but I'm definitely not gonna. I've actually never converted an Aptainer container to Docker, and I'm not definitely can't help you with that. And I'm not going to get into the details of differences between Docker and Singularity and Aptainer. Other questions? Okay, thank you. Please fill out the survey if you haven't already. Again, see you next week at the 1 to 4, the sort of regular time for the workshop series. And we'll be talking about single-cell RNA-seq. Okay, have a good rest of your day.